Generally refers to cellular biology and laboratory techniques involving the manipulation and study of cells. Here is a general guide on how to work with cells in a laboratory setting. Cell culture. Cells are typically cultured and maintained in a laboratory environment. This involves providing them with a suitable growth medium, temperature, and humidity conditions. Cells can be obtained from various sources such as tissues, cell banks, or cell lines. Sterile technique. Maintaining sterility is crucial when working with cells to prevent contamination. It is essential to work in a sterile laminar flow hood, wear appropriate personal protective equipment, lab coat, gloves, mask, and use sterile instruments and consumables. Passage and subculturing. Cells have a limited lifespan and need to be regularly subcultured or passaged to maintain their viability and prevent overgrowth. This process involves detaching the cells from the culture vessel, usually by using enzymatic or mechanical methods, and transferring them to new culture vessels. Cell counting and viability assessment. Cell counting is performed to determine the number of cells in a given sample, which helps in standardizing cell-based experiments. Various techniques such as manual counting using a hemocytometer, automated cell counters, or flow cytometry can be used. Cell viability assays are also performed to determine the percentage of live cells in a population. Transfection and transduction. Genetic manipulation of cells can be achieved through transfection or transduction. Transfection involves introducing foreign genetic material into cells using techniques such as lipid-based transfection reagents, electroporation, or viral vectors. Transduction utilizes viral vectors to deliver genetic material into cells. Cell differentiation and induction. Cells can be induced to differentiate into specific cell types by exposing them to specific growth factors, chemical compounds, or culture conditions. This is particularly important in stem cell research or when studying cell development and specialization. Microscopy and imaging. Microscopy techniques are commonly used to observe cells and their internal structures. Light microscopy, fluorescence microscopy, confocal microscopy, and electron microscopy are some of the techniques used to visualize cells and their components. Cell-based assays. Cells are frequently used in various assays to study cellular processes, such as proliferation, apoptosis, migration, and protein expression. Examples of cell-based assays include METET or XTT assays for cell viability, ELISA for protein quantification, and flow cytometry for cell analysis. Molecular techniques. Working with cells often involves extracting and analyzing cellular components, such as DNA, RNA, and proteins. Techniques like PCR, qPCR, Western blotting, and RNA sequencing are commonly used to study gene expression, protein levels, and other molecular processes. Data analysis. Finally, data obtained from cell experiments needs to be analyzed and interpreted. Statistical analysis, data visualization, and other bioinformatics tools may be used to draw conclusions from the experimental results. It's important to note that the specific techniques and protocols may vary depending on the type of cells, research goals, and laboratory expertise.